Madame Bruno, juste allumer votre micro.
Oui, ça serait une bonne idée. Alors, bonjour tout le monde. C'est vraiment un honneur et un privilège de vous présenter notre conférencier d'honneur de ce matin, le Dr. Michael Houghton. So, Dr. Michael Houghton is the Li Ka-shing Professor and holder of the Canada Excellence in Research Chair in Virology. He's the director of the Li Ka-shing Institute of Virology at the University of Alberta. As you know, Dr. Houghton is the co-recipient with his colleagues, Dr. Alter and Rice, of the Nobel Prize of Physiology or Medicine in 2020 for their seminal work that led to the discovery of HCV as the causative agent of non-A, non-B hepatitis, which I remember myself. Previously, together with Dr. Kuo and Dr. Xu at the Chiron Corporation and Dr. Bradley at the CDC, his laboratory discovered evidence of the Hep C virus. This work has a major impact as it led to the development of screening tests that has since virtually eliminated hepatitis C contamination of donated blood supplies in many countries, including Canada. Further, Dr. Houghton and his collaborators also linked Hep C to liver cancer, and his laboratory was also first to characterize the viral hepatitis D genome and human beta interferon gene. So his work is just remarkable and his trajectory is very special. But when you think about it, from the discovery of Hep C in 1990 to the development of the screening, fast forward in 2020, efficient antiviral drugs are now available, and the eradication of Hep C as a public health threat throughout the world is a goal for 2030. This is like going full circle in a way. Well, his work was not done yet. The Hep C pandemic still kills hundreds of thousands of people every year. And it's a pandemic, a word that is too familiar these days. Many countries and places in the world are simply not able to provide care and treatment, even if the drugs are made available. And vulnerable populations are still getting infected and not linked to care in Canada here. So Dr. Houghton moved to Canada in 2009 to work on an HCV vaccine. Many thanks to his good friend and longstanding colleague, Lon Terrell, to having convinced him to trade the California weather and palm trees for Edmonton chilly winters. Michael and I has been working together since, along with my close colleague Nagla Shukri at the CRSHN and many other researchers in the CanFC CIHR network. He's a tremendous inspiration for all of us and so generous with all the CanFC trainees. Michael is a wonderful colleague, always curious and interested in how his work impact people. In a way, you know, we work at the extremities of the research chain, him from his lab and me with the people in the streets. And Michael has such an extraordinary wide-angle vision and remarkable capacity to bring people together and inspire teams from different disciplines to work for a common goal. Michael, thank you so much to have accepted our invitation to present this morning. Please welcome Michael Houghton.
Thank you very much, Julie. Thank you for the invitation to speak today. And I'd like to thank you for all the wonderful work you've done in harm reduction in people at risk for HCV, HIV, and hepatitis B. That is tremendous work, and I thank you so much for what you've done and are still doing. And it's a pleasure to have worked with you. So thank you again. I need to get over to my slides. Always a challenge. Here we go. Share screen. That was easy. So thanks again to Julie and to Eric for speaking to you today. And as you can see, I'm going to talk about Hep C, vaccines, COVID, and offer some thoughts on how we should prepare ourselves for the next pandemic, which is sure to come. So Hep C is still a very big problem. World Health Organization estimates there are 2 to 3 million new infections around the globe every year. They estimate there are around 400,000 HCV-associated deaths every year around the world. And fortunately, not as severe as COVID, but nonetheless, it's obvious that Hep C infection is a huge epidemic in itself. And as Julie said, we have very good antivirals now as a result of the academic and private sector collaborations. We have good diagnostics to prevent transmission of Hep C in the blood banks around the world. What we need now is a vaccine, I believe, to really curb the epidemic. The US CDC estimates there's around 20 to 30,000 new infections of HCV every year in the States. Around 2/3 of these occur in people who inject drugs, sharing needles. Unfortunately, is a very efficient way to transmit hepatitis C. And as you're aware, we've had a growing number of epidemics in opiate users in USA and I believe in Canada as well, for sure. So in Canada, the Public Health Association estimates there are up to 12,000 new infections of HCV every year in the country. Once again, 2/3 or so occur in people who inject drugs. So I just wanted to introduce traditional vaccine technologies. Perhaps not everyone in your audience is familiar with what has been available to us up to COVID. So quickly, as most people know, smallpox was eradicated through the use of a live attenuated vaccinia virus. There are other examples of that for yellow fever. Mumps, measles, rubella is a live attenuated vaccine that protects our children, as is the chickenpox attenuated virus, rotavirus, which has been a very effective vaccine in developing countries to protect against the lethal effects of rotavirus, and of course polio, the Sabin vaccine. These are all good examples of live attenuated vaccines. The second category are vaccines where the virus is grown up in large amounts, purified, inactivated, and then given to people with an adjuvant to stimulate the immune response to the vaccine. And there are plenty of examples of this technology against flu, hepatitis A, polio again with the Salk vaccine, and rabies. I think in the last 30 years, the sort of fashionable vaccine, so to speak, became using recombinant subunit proteins, highly purified, non-infectious, with adjuvant. And we've seen some tremendous examples of how good these vaccines are against hepatitis B, papillomavirus, herpes zoster, shingles virus, hepatitis A, and also some bacterial vaccines use recombinant proteins like the whooping cough vaccine, which is part of the DTaP. And then just very briefly, there are bacterial toxoid vaccines where the lethal toxin is chemically inactivated, and that protects against diphtheria, tetanus, and pertussis also. And then a huge leap forward where people discovered if they put T helper epitopes conjugated physically to polysaccharides of bacterial capsules, that was a very good way to get protective antibodies against Haemophilus influenzae, meningococcal strains, Streptococcus pneumoniae, and so forth. So we have a good armory of vaccine approaches to deal with pandemics. For HCV, many years ago, when I was still in the States, we decided to go for a recombinant subunit vaccine where we express the two envelope glycoproteins in mammalian cells. We chose Chinese hamster ovary cell lines. To my knowledge, this is still the only vaccine for Hep C that has been shown to be protective in the fully immunocompetent chimpanzee model. Unfortunately, we lack good animal models for HCV, except pan-troglodytes, which is the common chimpanzee. So a lot of the work I did in the States was using that chimpanzee model. And it's important to understand what we're trying to do with a vaccine to Hep C, and also many vaccines, including COVID. We're trying to prevent disease. And with Hep C, disease is associated with the chronic persistent infection phase of the virus rather than a recent acute infection. Recent acute infections of Hep C are usually, almost without exception, subclinical. So we're trying to prevent the persistence, which can lead to cirrhosis and liver cancer and end-stage liver disease. You can express the E1 and E2 genes, the envelope glycoprotein genes, together in mammalian cells, and they are heavily glycosylated, N-glycosylated, as well as O-glycosylated. So you can see the large E2 glycoprotein here on a gel and the smaller glycosylated E1. They do associate together in a heterodimer. They are not secreted from the mammalian cell, so it's a difficult antigen to express at high levels. We have to extract the heterodimer from the lumen of the ER, but that is possible to do. And then in the States, we immunized, over many years, we immunized chimpanzees with the E1, E2, along with various adjuvants. And then after 3 immunizations on months 0, 1, and 6, approximately, we then challenged with either homologous virus or heterologous virus. And we challenged usually anywhere between 2 to 4 weeks after the final immunization. And this is a summary. What it shows is that if we challenge with homologous virus, the same virus that the vaccine was made from, we had 12 animals vaccinated. 5 of them were sterilized against infection, which was a surprise to me. I didn't think we could sterilize animals, but we did. 5 of them. These were the highest responders to the vaccine at the antibody level, and they were completely sterilized. The other 7 got acutely infected. But only 2 went on to chronic persistent infection, which is 17%. Of 10 unimmunized controls, 70% went on to chronic persistent infection. And we had statistical significance at preventing viral persistence in the homologous challenges. With the heterologous challenges, of 19 animals, all of them got infected. We didn't sterilize any of those, but only 3 went on to develop chronic infection, just 16%. Of 14 unimmunized controls, we saw 57% go on to develop chronic infection. So this was data compiled over several years. It also includes some of the NIH's unimmunized controls. And you can see that either with homologous or heterologous challenge, we saw a statistically significant decrease in chronicity levels, persistent levels of the virus. And if you combine the data together, it's very highly significant. Now, Hep C is actually more heterogeneous than HIV. There are 7 major genotypes around the world with hundreds of subtypes in each major genotype. So heterogeneity has always been an issue with Hep C, like it is with HIV. But we've shown that actually E1, E2 derived from a single strain can elicit broad cross-neutralizing antibodies. This is work we did with J.C. Mounier in Bob Purcell's lab nearly 10 years ago. We gave him 4 of our chimpanzee vaccinees, and he looked at the ability to neutralize different genotypes of HCV, 1A, 2A, 3A, all the way to group 6. So these are, I said 7 earlier, which is true, but the 6 major genotypes account for 99% of the world's infections. And to our delight, we saw that the antisera to E1, E2, once again from a single strain, could neutralize homologous a heterologous virus of the 1A genotype. 1A is the predominant genotype in Canada and in the US, but we could also neutralize genotypes 4, 5, and 6 very well. Genotypes 2 and 3, which do occur in Canada, they account for maybe 30 to 40% of infections. We were able to neutralize those, but as you can see, less well than the other genotypes. And my colleague, John Law, who I've worked with at the University of Alberta for 10 years, who's a really good virologist, he was able to show that in a clinical trial that we ran when I was in the States, it was E1E2 plus MF59 adjuvant. He looked at some of the high responders to the vaccine, and he looked at individuals 1, 5, and 7, and he looked at the ability of those recipients of the vaccine to neutralize the many genotypes around the world. And in, for example, number 5, we could see evidence that even though the vaccine is derived from a single strain, it can neutralize all the different genotypes to various extents. Once again, genotypes 2 and 3 were less well neutralized, although there is some variation. Individual number 7 neutralized 2A quite well, as you can see here in yellow. But the important message here is that even though it's a very variable genus, there are obviously highly conserved neutralizing epitopes in most of these viral clades and strains, which is really good news for vaccine development. And then one of our students, Jason Wong, published in 2014. Once again, he looked at the human recipients of the vaccine, and he looked at 5 recipients, V1 through V5, and he asked the question, does their antisera compete for the binding of highly cross-neutralizing monoclonal antibodies? And the antibodies are shown here, AR3B, 4A, 5A, and AP33. Before vaccination, you don't see any inhibition of binding of these monoclonal antibodies to E1E2. But after vaccination, we saw evidence for the generation of antibodies to each of these highly cross-neutralizing antibodies for HCV. You can see each of these individuals. V5 apparently induced less antibodies to these epitopes than V1 and so forth. But the point is they are there in the vaccinated individuals, which once again is very encouraging for vaccine development. The correlates of protection to HCV has been a complex issue, I would say, partly because it took a long time to develop neutralizing antibody assays for HCV. They didn't really become available until 10, 15 years after we had identified the virus. It's a very difficult virus to grow in cell culture. But I think today, the case for neutralizing antibodies in the protective immune response, I would advocate these examples. Dr. Moran, 8 years ago, showed that a neutralizing monoclonal antibody, a single one, can protect chimpanzees from infection. Secondly, Dr. Meuleman in Belgium took immunoglobulins from a chronically infected patient and showed that in a SCID mouse model, which has human liver growing, colonizing the mouse liver, this is a model developed by my colleague, Dr. Lorne Tyrrell at the University of Alberta with Dr. Knieneman and Dr. Mercer. So the Belgians used this model, and what they showed is that immunoglobulin from a chronically infected patient can actually prevent infection of that mouse model against homologous and heterologous challenges. And the heterologous challenges were very extreme heterologous challenges. So they were all of the genotypes that occur around the world. So this is, I think, persuasive evidence that antibodies really play a role in protection. Dr. Lavillette and Pesca and Kinchen more recently have all shown that neutralizing antibody titers correlate with recovery in humans Most humans develop persistent infection, but around 20% can spontaneously resolve the infection on their own. And neutralizing antibody titers do correlate with that recovery. They have the highest titers of neutralizing antibody. However, and I think this is the core of HCV, it's been shown by Dr. Kosei's group originally, and more recently by Dr. Peachman's group and Dr. Bankwitz, who published a very nice paper a few years ago. I think the key to Hep C is that antibodies, neutralizing antibodies, do provide protection, but the virus is protected somewhat against neutralizing antibodies. First of all, it has this very huge complex glycan shield, just like HIV does on its virion envelopes. But HCV has another trick up its sleeve, and that is it associates with human serum lipoproteins, and in fact is secreted on the apolipoprotein pathway. And it's been shown by Dr. Kosei's group and Dr. Peachman's group that these lipoproteins, they don't completely prevent the action of neutralizing antibodies, but they do reduce the action of neutralizing antibodies. And I think right there is why Hep C is a persistent virus, as opposed to Hep B, which usually is not, and Hep A, which never is persistent. I think it's because the virus has evolved methods to give partial protection from neutralizing antibodies. There's a strong case for cellular immune responses in the protective immune response to HCV. And this really, the best evidence comes from very nice definitive studies in Chris Walker's lab in Ohio State, where Andre Gakui and our own Nagla Shukri, who's at the University of Montreal and who we have a fine collaboration with, they showed that if you depleted chimpanzees of CD4 T cells or CD8 T cells using an antibody depletion method, you actually do prevent spontaneous eradication of the virus in challenged chimpanzees. So that was very clear cut. So we know the cellular immune response is definitely important. And so what we're doing at the University of Alberta is using E1E2 as a source of cross-neutralizing antibody responses, but also my colleague Amir Landi, who's a cellular immunologist, we've decided to focus the immune response also on small synthetic peptides that contain highly conserved CD4 and CD8 epitopes. So our vaccine is a combination, obviously, of a glycoprotein made in CHO, as well as synthetic peptides. And we're going to try and use 4 peptides. This peptide, for example, has 10 highly conserved CD4 epitopes, conserved amongst the whole genus, 4 CD8 epitopes that are presented by multiple MHC class 1s. And then we have other peptides less abundant in epitopes as peptide 1, but we're adding these to give wide coverage of both CD4 presentation and, importantly, CD8 presentation through various MHC class 1 alleles. And we're using an adjuvant that I've used before that I've shown is capable of priming strong CD4 responses, but also strong CD8 cytotoxic lymphocyte responses. So we're hopeful that this is going to give us the kinds of immune responses we want in humans. So where are we with this vaccine? We're currently manufacturing it actually at the University of Alberta. We've got this wonderful GMP facility that's required for clinical trials. We certainly feel we can provide enough for all the high-risk groups in Canada. These would be young intravenous drug users who are seronegative for HCV. I think if we can show efficacy in a phase 2 trial, our regulatory consultants advise us we will be able to roll out the vaccine immediately into high-risk people who inject drugs. And maybe we'll be able to do that around in 5 or 6 years' time. And this will dramatically lower the national incidence of infection, I hope, which is currently up to 12,000 infections per year. And if you look at the health economics, I think we can do that in these high-risk individuals in Canada for a cost of around $20 million. And if you look at the other scenario where you let all of these people become chronic carriers and then you have to treat them with the expensive antivirals, that will cost in the region of $1 billion. So there's apart obviously from the medical advantage, there's a huge pharmacoeconomic argument to roll out a vaccine in high-risk people in Canada. Moving to COVID, I think what's been wonderful is COVID vaccine approaches, the new ones, the new technologies look like they're working, touch wood. So far, it's very encouraging. And as you know, and I'm sure you've been discussing this morning, there are two kinds of new vaccines that are being approached with COVID. The technologies have never been used in approved vaccines before, but the data so far is looking very promising. And one is the use of replication defective adenoviral vectors to deliver the spike protein of COVID. So Oxford really pioneered this approach thanks to Dr. Adrian Hill's work on malaria, and Dr. Gilbert at Oxford has really taken that up for COVID and partnered with AstraZeneca. But we've got two other companies working on this, J&J in the States and CanSino, which is a Chinese company, but is working closely with the NRC in Ottawa. And then, of course, we have the lipid RNA nanoparticles from Moderna and BioNTech, who each have their own big pharmaceutical partner, Lonza or Pfizer. And this is very interesting technology that looks like it's going to transform the vaccine industry, certainly has the capability of transforming the vaccine industry in the future. They use modified nucleosides to inhibit the innate immune response. If they don't do that, then expression of the RNA molecule is inhibited greatly by the innate immune response. And so they modify some of the nucleosides to prevent an overwhelming innate immune response. They add a cap structure at the 5' end to increase translation efficiency, and then to stabilize the RNA and also to enhance its uptake by antigen-presenting cells, they formulate the RNA into lipid nanoparticle formulations around 140 nanometers in size. So to the immune system, it looks like a virus, an RNA virus, which is good. There are other leading approaches, for example, Novavax, Medicago, and Sanofi GSK. They're all going the protein subunit approach using powerful adjuvants. This is also looking very promising. And then Inovio are going the route of DNA vaccines. DNA vaccines on their own don't work in humans, we know that, but if you find some way to enhance the delivery, e.g. by electroporation, literally electrophoresing the DNA into the muscle of the arm, it's also looking a very promising method. So the Moderna vaccine, as I'm sure most of you know, comprises a RNA molecule with the modified bases, the cap structure, formulated into lipids, but it encodes pretty much the full-length spike protein of the COVID virus. And it's been shown by Dr. Walls and others this year that that full-length spike protein forms a trimer, shown here, embedded in the virion lipid membrane. And these spikes stick out from the lipid membrane in the form of a trimer. And this green domain is the receptor binding domain. It's very interesting that in the trimer, only one of the trimer molecules has the receptor binding domain available. The other two have the receptor binding domains pointing downward. They're not accessible to the receptor. There's a very interesting structure of this trimer molecule, it really is. But anyway, one of them has the receptor binding domain, and the receptor binding domain is in the S1 part of the spike in the N-terminal region shown here schematically. It's unfortunately not to scale, I apologize. It represents about 300 amino acids, so it's about half of the S1 domain, as it's called. In natural infection, you get cleavage here in the ER through a furin protease cleavage site, and you get formation of S1 subunit and S2. So Moderna have also expressed the full-length spike in their RNA vaccine, and as we've learned every day on the news, it's an interesting way to read science. You just pick up the news every morning and you find the latest development. So it's unpublished yet, but it's been reported to be 94% effective at preventing disease. Now, it's a 2-shot vaccine, I believe week 0 and I think week 3. And then they've been following up the cohorts. So there's around 40,000 people in that trial. Half of them get placebo, half get the vaccine. They've been following them up for anywhere between 1 or 2 weeks to 1 or 2 months. So it's still early days, but at this early point, they're seeing 94% effectiveness of the vaccine, which is really, really encouraging. Also, BioNTech, a German company who is partnered with Pfizer, they have a similar kind of RNA vaccine. I believe it's different RNA bases to Moderna and different lipids to form the nanoparticle, but it's the same concept. And they've reported recently also a very high efficacy in the short-term follow-up after a 2-dose immunization regimen. They're reporting 94% efficacy also. And both Moderna and BioNTech are finding efficacy in elderly people, which is also fantastic and very encouraging. You know, both RNA vaccines seem to be well tolerated. You get typical vaccine effects. If you get a powerful vaccine, I had the Shingles vaccine recently, a few weeks ago myself, the new one from GlaxoSmithKline, which is extremely effective and highly recommended for elderly people where the risk of shingles is very high as you age. So typically, the RNA vaccines are giving injection site pain for 1 or 2 days. You get a bit of fever for 1 or 2 days. You might get a headache for 1 or 2 days. You feel under the weather, but that's really typical for many vaccines that we get, including the shingles vaccine. And I read on the newspapers this morning that in the UK have approved the BioNTech Pfizer vaccine for human use. And that just happened this morning, and they're about to roll out the vaccine into the high-risk groups within a few weeks. You know, obviously, we still need more information. As I mentioned, RNA vaccines have the potential to transform how we deliver vaccines, viral vaccines, bacterial vaccines, and parasitical vaccines. But there are some important questions left, of course. We don't know the duration of immunity yet. We know that there's short-term immunity, but obviously we need long-term immunity. To do that, it wouldn't be surprising to learn that we need additional booster shots, perhaps annual booster shots against COVID to maintain protective levels of antibodies and T cells, effector T cells. We don't know yet the potency of this type of vaccine against others. We've seen data from Oxford and AstraZeneca that their vaccine is anywhere between 60 to 90% efficacious. They got different data, different results from 2 different phase 3 clinical trials. But we don't know yet how the RNA vaccines will stack up against protein subunit vaccines with adjuvants. There are issues of cost of goods. When you have to vaccinate the world, cost of goods becomes really important. And as you know, probably, there's a cold chain requirement for the RNA vaccines, which is quite a severe one for the BioNTech Pfizer. That requires -70. Moderna's and Lonza's requires -20. However, when they're thawed, what I'm reading in the newspapers is that Moderna is then stable at 4 degrees centigrade for 30 days, whereas the BioNTech vaccine, once it's thawed from -80, it's only stable for 1 week. Doesn't mean we can't use the Pfizer vaccine, not at all, but it just makes it somewhat harder for delivery. And the adeno vaccines can be stored at 4 degrees, so they are much easier to deliver. What we've done in our institute is sort of have a backup vaccine strategy. When we started this, we didn't know if the RNA vaccines would work, and we didn't know about adeno vaccines. So what we decided to do is to focus on the receptor binding domain of the spike protein. And we've expressed that in mammalian cells, and we've provided an adjuvant, and we've immunized mice. And John Law and Mike Logan have been at the forefront of this. And we're looking at not a very nice graph. For some reason, it's like the x-axis, but we're looking at the ability of mouse antisera to neutralize the infectivity of COVID pseudoparticles expressing the spike protein. So this is a surrogate neutralizing assay. Dr. Tyrell recently got data showing that indeed we can neutralize the infectivity of the virus itself in cell culture. This happened just a few days ago. I don't have a slide of that yet. I think Lawrence Lab is just looking at other dilutions, but this is the pseudo-particle assay. And you can see that at dilutions up to around 1,000, up to 1350, you can dilute the mice antisera up to 1,000-fold, and you can essentially completely prevent the entry of COVID pseudoparticles into cultured cells. So I think we've shown that this is a valid approach, and another group from China has also presented persuasive evidence that a vaccine targeting the receptor binding domain, which interacts with the angiotensin converting enzyme 2 receptor on human cells, this is a valid approach, and it's working well in our hands. And one of the reasons we chose it is that that small protein domain, which is about 300 amino acids, we think we can express it at the levels needed for national vaccination. So a couple more questions before I stop. I think there's been a lot of lessons that COVID has taught us. And I think one of the questions has been, would a vaccine stockpiled against the coronavirus, that the SARS coronavirus that emerged in 2003, if we had a vaccine to that stockpiled, would that have prevented the COVID-19 epidemic? And I think the answer is it would have really helped and would have stopped a lot of disease and a lot of fatalities, because this is work from Dr. Walls' group, published in Cell earlier this year. He immunized mice with the SARS coronavirus 1 spike protein with adjuvant. And then he looked for the ability of the antisera to neutralize the coronavirus 1 infectivity in cell culture. And you can see here, this is a control where control antisera is used, and you get 100% entry of the pseudoparticle from coronavirus 1 spike into cells. But all the mice that he looked at, 1, 2, 3, 4 mice that he immunized, completely prevented entry of the coronavirus 1 pseudoparticle into cells. Then he asked the question, what about coronavirus 2, the COVID virus? This is the control, and you can see those 4 same mice had very potent activity at preventing the entry of these HCV pseudoparticles, sorry, these coronavirus 2 pseudoparticles into cultured cells. Not quite as good as against the homologous coronavirus in 2003, where it was completely prevented. But here, against COVID virus pseudoparticles, Sorry, this is actual virus we're looking at, not pseudoparticles. He showed that essentially the vaccine to the 2003 strain of coronavirus inhibited by 90% the infection of cultured cells by the COVID virus. So this is a big lesson, I think, we have to learn from this terrible pandemic. So how should we prepare? This is my final slide. How should we prepare for the next pandemic? What has COVID taught us? I think the first thing it's taught us is that we should stockpile vaccines. Don't stop manufacturing vaccines and storing them because the epidemic declines and disappears. This has happened with several epidemics. West Nile virus, for example, was another example. Flavivirus, SARS 2003 certainly is a classic example, where if we had have manufactured vaccines ready for human use and stored them, the sobering thought is that we could have prevented much of the morbidity and mortality of COVID-19 around the world. And certainly recombinant protein vaccines, once you've made them, they are very stable. The E1E2 that we made in the States was stable for 10 years or more at -80. It's highly purified, so there's nothing going on at -80 with those proteins. I think secondly, I think COVID is a wake-up call to the human race, and I think governments, I would recommend governments responding by increasing the funding for both basic research and translational research in infectious disease. This will not be the last pandemic. Anybody in infectious disease knows that. We're likely to get a pandemic from an influenza virus or another coronavirus or who knows what in the future. I think we need to boost our funding for basic and translational research in infectious disease. I think another thing that COVID taught us in Canada is that we don't really have, to my knowledge, we have some small pharmaceutical vaccine companies. We have Sanofi Pasteur in Toronto who are building a big plant targeting mainly childhood vaccines. We don't have the manufacturing capacity that, for example, the US has and the Europeans have. So we need to increase our manufacturing capabilities so that in future pandemics, we have the ability to make RNA and other vaccine types, including subunit protein vaccines, so that we're not so dependent on other countries for vaccine supplies. You know, Canada has bought a lot of the leading vaccines in advance of the clinical trials, which is great. But we're not sure yet when we're going to get those vaccines available to Canadian citizens. So we need to become more independent at the manufacturing level. And I think finally, we do need to sit down across the world and we need to discuss what can we do in terms of public health measures to try and minimize the risk of future pandemics. As you know, these viruses often occur, influenzas, often the reservoir are the aquatic bird population. Coronaviruses have different original species that they infect, and there's obviously good evidence that COVID came from bats. So we need to figure out a way sensibly to discuss how we can have some public health measures, probably fairly simple ones, to reduce the likelihood of future pandemics. So I end my talk. I need to acknowledge my many colleagues at University of Alberta for the last 10 years that I've been working with on the Hep C vaccine, more recently on the COVID vaccine, all really talented young people. And Lorne Terrell is a very talented elderly person whom I'm indebted to. He's been a wonderful collaborator. And I'd like to thank also Julie and Nagla. You've been wonderful collaborators on the Hep C program for many years. Julie does this wonderful work in harm reduction, and I really so admire what she does to prevent infection in people who inject drugs, HIV, HCV, and HPV. And Nagla is a fantastic cellular immunologist that we gain from working with and enjoy working with. So thank you very much for your patience.
Well, thank you very much, Michael. If I had a applaud here, we would hear a lot of applaud, I'm pretty sure. And thanks for your kind words. That was a very interesting talk, and I think it gives a very wide and interesting perspective on vaccine. I have a few questions, so I'll read them to you.
Sure.
First, from Petronella and Kuta. So thanks a lot for your presentation. Multiple viruses, polio, noroviruses, depend on a specific bacterial niche to infect target cells. Do you think there is a need for a special bacterial environment for HCV to grow in vitro?
Interesting question. To my knowledge, there's no evidence for that. So I don't think so. No, I've never seen any report on a requirement for bacterial co-infection for Hep C. So I don't think so. No.
Thank you. And the other question from Jean-Charles, who is also thanking you for your impressive presentation. Do we know whether or not CHO cells, not the CHO developed vaccines, just the cells, can induce metastasis in SCID mice? And there is a second part of it is, can the current vaccines being developed protect against most all SARS-CoV-2 variants, including those variants with mutations in S protein coding gene?
Yes. Yeah. So the last question, a lot of work that's been published by many of the vaccine groups around the world, they have shown that the neutralizing antibodies generated by their vaccine do neutralize all the strains of COVID around the world. I'm thinking primarily of the Sinovac inactivated virus vaccine. They published very nice data showing that they can neutralize all the strains of COVID. And some of the other groups have published similarly. There is an issue with this virus in Denmark that passed from humans to minks. It adapted to the mink by making mutations in the spike and then came out and reinfected humans, or I should say infected other humans. There are some mutations in that spike protein, but there is no data that I've seen on whether or not it is neutralized by the vaccine-induced antibodies. You know, that mink virus that reemerges in humans, it still uses the ACE2, angiotensin converting enzyme 2 receptor in humans. And so I will be very surprised if vaccine antibodies don't also neutralize it. But that said, it's a possibility they may not neutralize as well as some non-mink viruses. But I've never seen any data on that. I'm trying to get hold of that mink virus from Denmark. Haven't been able to get it yet, but so we have to wait and see. But I would be very surprised if there's not neutralizing activity against the mink virus. And the first question, I think you were asking in the mouse model for Hep C, where you can grow the virus in human liver cells that colonize the mouse liver, do you get hepatocellular carcinoma? Well, you know, that mouse model is a delicate model. You can keep those mice for months, maybe a year or so, but I think, in other words, the experiment hasn't been done. They haven't been left long enough, and of course, mice have short half-lives, short lives. So I don't think so. I can tell you though, in chimpanzees, I only know one case of hepatocellular carcinoma in a chimpanzee in all the studies that have been done around the world for the last 30, 40 years. So there have been reports in transgenic mice expressing the core protein of Hep C. Frank Zari, his group, reported some hepatocellular carcinoma developing in his mice. But I think the levels of expression in his mice were much higher than what we see in Hep C patients. So I suspect most of the hepatocellular carcinoma in humans by HCV is induced as a result of the immune response and persistent inflammation, necrosis. And I think mainly that's what's responsible for hepatocellular carcinoma from HCV. You can't completely rule out a direct viral effect, but I think there is one for Hep B. But for Hep C, I think mostly it's indirectly as a result of immune damage over the course of many, many years.
Thank you very much. So I get, is there any other questions? I want to thank you again. And I think I want to end by saying that if COVID, despite everything that's happening, is putting more focus on developing more vaccines for the other pandemics and the ones to come, I think it would be at least a secondary outcome that would be positive for that horrible pandemic.
Yes, I agree with you, Julie. I think COVID is teaching us some lessons on how to develop all the other vaccines. So I totally agree.
And hope that the funding will come with it.
Yes, yes, we need to get more funding for infectious disease. I think that's very clear. And yeah, look forward to continue work with you, Julie. And thanks very much for inviting me. It was a pleasure to contribute to you. Yeah.
It was a real pleasure. Thank you so much.
Thanks, Julie.
So I'll just end up by saying as well that at the audience that le symposium va continuer à 8h30 demain matin avec 2 sessions, une sur les cohortes et aussi sur les essais cliniques et aussi l'impact de la COVID sur les personnes vulnérables. Alors j'invite tout le monde à être au poste demain matin à 8h30. Merci beaucoup.